webinar. All right, welcome. Today is Tuesday, October 27th, 2020. Thank you all for joining us today for the Soil and Water Conservation Society's Technical Workshop webinar. This webinar will cover a recent project from Michigan State University's Institute of Water Research. The Institute of Water Research is working with several partners, including the Michigan chapter of the Soil and Water Conservation Society, to address water quality concerns. The Soil and Water Conservation Society's mission is fostering the science and art of soil and water conservation in Michigan and the Great Lakes region. Next, on to our agenda. During this webinar, Jeremiah Asher from the Institute of Water Research will provide a project overview. Jeremiah will then present a recording of floating wetland treatment of subsurface drainage. A Q&A session and update will immediately follow his presentation. Now for a few reminders. As previously stated, each project presentation will be followed by a Q&A session and an update. The questions can be submitted at any time using our Zoom toolbar. All attendees' video and audio features are disabled for this webinar for privacy concerns. This webinar will be recorded and available through the Michigan Soil and Water Conservation Society's website. Attendee names will not be called out during the Q&A session and will not be included in this recording. Now for a few introductions. My name is Katie DeRocha. I'll be your webinar host for today. I'm the Vice President of the Michigan chapter of the Soil and Water Conservation Society, and I am a soil conservationist for the Kent Conservation District located in Grand Rapids, Michigan. Next, I would like to introduce our speaker, Jeremiah Asher. Jeremiah is the Assistant Director at the Institute of Water Research and has an extensive background in the Geographic Information Systems, GIS, Decision Support System Development, and Natural Resource Management. His research focuses on web GIS application development. He has developed several nationally awarded assessment and planning tools for local and state governments. His most recent activities are focused on developing sensors for managing water and edge-of-field floating wetland research. At this time, we will move on to the recording of floating wetland treatment of subsurface drainage. Jeremiah, thank you for joining us today, and now on to your presentation.
Well, today what we're going to be talking about is a wetland treatment system that we have set up here that we're doing some research on. This is a two-stage wetland treatment aimed at trying to reduce nutrients that's coming off of agricultural fields. We put this in, it was constructed last October, and then it kind of sat through the winter time, and then in the spring we put out some equipment that we'll talk about here in a little bit to start testing. And mostly this summer we've just been trying to calibrate the equipment, understand better how the system is working, and then starting now in October we'll move forward to the beginning of our actual data collection for this first coming year. So that's kind of the background of the project here. And just to give you some information about how this all got started and funded, it kind of all the stars aligned a little bit. The Army Corps of Engineers was interested in looking at wetlands to reduce nutrients off of agricultural fields. And they liked the idea because wetlands had a little bit more permanency than maybe infield practice where commodity prices might change and that practice might come out of the field. So it's a little bit harder to remove a wetland once it gets established. So they were interested in that, and so we were able to get some funding to construct that, but we didn't really have funding for doing the monitoring. So around the same time, Dennis and his group put together a proposal to NRCS for a conservation innovation grant to look at low-cost monitoring equipment. So he was looking for sites, and we were looking for monitoring to look at the performance of these. And so we were able to kind of put this together and get the monitoring equipment out here so we could test that and then also test the system out to see how it was doing. And then, as you know, we're up in Michigan State, up in Lansing, and so it's a bit of a drive to come down here to collect samples every day. So we actually work with Adrian College, and Dr. Martin and some of his students are helping to collect water samples. So they come out here every 48 hours. They'll collect from these 3 different monitoring stations here, and then they take it back to a lab at Adrian, and they do the analysis and then send us the information. So that's kind of how the whole system works here. So, give you why are we here, and I don't mean existentially, but why are we here at this location? So if you remember the commercial in the 70s and early 80s with Life cereal, and you know they had Mikey, and you know the brothers would say, yeah, give it to Mikey, you know he'll try it, and that's kind of like Blaine Baker, whose property we're on now is, you know, Blaine is our Mikey in a sense. So you know, we really thank Blaine for being able to come out here and do this research. He's made these research projects available to a number of different researchers who are trying different conservation practices and doing different research. And so we wouldn't have been able to do this without Blaine's help in setting this up. In addition to that, many of you have seen some of this. This was published out of the Journal of Great Lakes Research. And this is this is Lake Erie, and this is showing total phosphorus coming off of the landscape. And so in the western Lake Erie Basin, the majority of this 89% is coming out from non-point source. So we're interested in looking at what ways can we help to reduce non-point source pollution from getting into waterways and into the basin. In addition to that, there was some additional research that came out looking at the pathways off of agricultural fields for how how nutrients, particularly phosphorus, is getting in to streams. So this is work that was done by Kevin King, and essentially I'll try to summarize what these are. I tried to highlight these in red here, but across all seasons, the primary sources or pathways for nutrients, phosphorus getting in or off the field was through subsurface drainage. And this is both the particulate and the soluble portion. And also annually, the greatest contribution was coming from subsurface, and then in the critical months, that was also coming through subsurface. So we wanted to try to put together a system that could help to reduce those coming through subsurface. And another reason why we were here, so this is Lake Erie. This is the River Raisin watershed, which you guys are all in right now, right here at this little dot. That's where we're standing in this field. And this was some modeling that was done by U of M that used the SWAP model to look at the concentrations of dissolved reactive phosphorus by sub-watershed in the River Raisin. So these 2 areas here are what they consider hot spots. These are the highest contributing, according to the model, contributions of dissolved reactive phosphorus coming into the system. So we wanted to be in one of these places that we could try to focus some of our efforts. So again, that's why we're here. So let me talk a little bit about how this system works. So this is just an overview of what a diagram of this system. So we have this monitoring structure over here, A, B, and C. There's 3 monitoring structures that we're looking at taking samples from. And basically what we have is from this field up here, we have water coming through a tile drain, and it goes into this control structure, and we have equipment in there that looks at what are the flow and what is the nutrients that's coming off the field. Then we come into this first treatment cell, which is a surface treatment. So what we're using for surface treatment is we have floating mats that have plants that are in those, and I'll pass some of these around, and I'll explain a little bit how these work. But these are little plant holders that snap into this mat, and you can see these right here. This is an example of the mat. They snap into that mat, and then the root systems grow through, and as the water levels change, the mats just float up and down so they can fluctuate with the water level. And there is a lot of fluctuation in these, and I'll talk about that again in a little bit. But what those plants do is they take up nutrients from the water, and then what we can do is harvest those at the end of the year and then reapply those back in the field or recycle them some other place. And then the second cell, so then we come through here and we monitor that, so we take samples to see what's the difference between what came out of the field and what happened in this first pond to then going in the second pond. They then come over and they go through a subsurface treatment. And with the subsurface treatment, we have a sand and limestone mix there where phosphorus will absorb to minerals and stuff in that filtration. And so that'll pull additional nutrients out. And what we try to do with the treatment, this combination or treatment train, is that we can remove kind of in this first stage some initial phosphorus and also let some sediment settle out of here. And then as that will extend the life of that second cell. Eventually, these systems will saturate, but we're hoping that we can extend the life of this system by using that first one. And then after it filters through there, it goes over, we monitor it again, and then it continues out to the drain. Originally, when we came here, there was a tile drain that just that opened up right here to the surface, and it just ran across the wooded area here for 30, 40 yards before it just went off into the field. So we changed some of that here, and we added in some wetlands. Actually provides a lot of habitat for frogs and different things we've seen over the summer. So a few of the things that we're testing with this system is, one is we want to know how much, what kind of nutrients the system is removing, both from the first cell and the second cell, and then as a combination system. In addition to that, we want to test the sizing. So previous publications have said about 1% to 2% pond size or wetland size to the area that you're treating. And so if we followed that, we would be about 4 times as large for this system. And it's hard to get that much land in a farm field to work with. But, so we tried to go as small as we could. And so that's where we came up with this design. And we were hoping that, you could even take these and make them skinnier and longer and put them maybe along a buffer strip or something in a farm field. So that's why we were testing the size. We also want to test different plants. So we have 4 different, and I left the labels in the car. 4 different plants that we are testing, and we measure the weight of those and then digest those to see how much phosphorus they begin with, and then at the end of the season here we'll see how much phosphorus they've removed from the system. And we want to see which of those are performing better, and then we'll add additional mats out here and more of those types of plants. And we'll continue to test those for a couple years. But what was really interesting is when we put these out here, and you can see these root masses here are really significant. Some of these, I think this one here, if I pull that out, and we can look at it in a little bit if you want, but it's about 24 inches long when you pull the whole thing out. And what those do is, underneath those mats, they all kind of intertwine together, those root systems, and they hang down in the water, and they grab and almost act like a flocculent, where they grab sediment and other suspended solids and take those and pull them together and drop them out, settle them out at the bottom. So that helps to filter that water through there as well as taking up additional nutrients. So this has been really interesting. Matt, who's snapping photos over here, was with us this summer when we put these in June. So we basically took all these and put them in these little plant containers here. None of the roots when we started were through these plant containers. And this is what's happened since we've had them in June. So we've been really happy how that's come along. And we've been able to take pictures. We have video and snapshots here mounted, so we can look at these ponds at all times from up in Lansing, see how the plants are progressing and if there's any trouble with the pond system. So this is just to give you an illustration of what we're actually treating here. So you can't see this, but these are you guys here. These are the people. But here are the 2 ponds. And where we first came in off the road this way, this is all tile drains. So there's about 7.5 acres that way that's just tile only. And then right above us here, if we look straight back, you'll see there's a little mound of, looks like grass or something that's back there. That's this thing here, which is called a wascob. That's a water and sediment control basin. And what that does is, it's pretty common in this area. There's a lot of kind of rolling, sloping hills. And in order to help control erosion, they put they're basically berms here, and they collect surface water runoff, and then there's a pipe, which is like a surface riser, and the water collects there and then runs into that pipe and then connects with the tile drain. So this 8.8 acres above us is actually a combination of surface and subsurface drainage. So that all collectively comes through into this. And as you know, this spring we were significantly wet, and so calculations for trying to do sizing for this were a little off. We had one day that we had about 550,000 gallons in a day that came through the system. And so for the official international calculation, that's 124 above ground pools of water that came through this system in a day. This one can handle about 30,000 or so gallons. And so we went to the capacity of that first cell. That was the day, and you can see the top of this overflow part here in yellow was almost full, and we were coming out to the end of the tile, which I'll show you here in a minute. So there was a lot of flow that day. We're looking at ways that we can modify this system to either do just tile drain, so only tile drain between the two systems. That'll remove some of that peak flow. Or we might try to do something where we put in a control structure and we put an automated gate. And so this is another thing that we're testing that you can pass that around. That's something that we built that we're using to look at surface water. You can open it up if you want. And we'll look at the surface water depth here and try to calculate where that depth is, and then we can open and control gates so that we put enough water in there, and then we'll just divert the rest of the water off if it's a peak flow that's too high for us to manage. So I'm trying to think if there's any other pieces here. So let me talk a little bit about what we found so far. And this is, please draw no conclusions from this because it's just in our calibration period. At the beginning of the year, we got about 75% reduction of phosphorus. We're measuring currently dissolved reactive phosphorus nitrates coming from these. We'll be adding some additional, the whole suite will do total phosphorus, ammonia, and total nitrogen sampling, but this summer we've only been collecting those two. But we've had reductions of about 75% coming from this through the back part of the system. For one of the first early months that we observed. Then the second system clogged. So there's a filter fabric in there, and we think that some fines from the sediment got in and clogged those systems up. So we're actually in late October, November timeframe, we're pulling that fabric out of there and putting in a more porous mixture so that second system will translate through faster. So this summer, after that first month, we really don't know what was happening with it because we couldn't monitor it. In March, we saw a 50% reduction of nutrients coming from here to that second cell, but there was nothing in the pond. So, your conclusion, a false conclusion would be, hey, we can just dig some holes and, collect some water coming out of pipes and we would have some reduction in nutrients. But what we think was happening in there is during that time frame that there was the soluble portion of phosphorus was attaching to iron and other things and falling out and precipitating in part of the sediment at the bottom. So that's kind of being held in there, and it changed what we were seeing off of that, coming off that first cell. Then in April and May, we actually had like a 10% increase here, and then a 20% increase. So we started to see it increasing here. Anybody have any ideas why that might occur? Well, there was some releases, some possibilities, and then we also overlaid evaporation off the pond. And so we actually start to see evaporation starting to spike up in those months, April or May more than May. And so what we did is we saw that some of the water was starting to evaporate, which would change the concentration here. So I say all those things and that their systems are very complex. And for us, this first year of just calibrating and understanding flows in different parts of the system are really important so that this next year coming in, we'll have a better idea of how the system's actually performing. When we put the plants in June, even though we continue to see increases in evaporation rates going from May to June, we actually saw about a 15% reduction of nutrients coming from this first cell. So we feeling good that the plants are taking up and doing some of the part that they're supposed to do. And moving into this next year, we'll have a better idea of how those are performing. One last thing we are testing with these plants is hardiness as well, so we want to leave some of them in here and see how they do over the winter time and if some of them reestablish themselves in the winter. And so we're part of a new funding that we got from NRCS is to continue to look at these different types of plant species and which ones do well in cold climates so that you can leave them out, you know, year after year and maybe just harvest the top parts and let the bottoms continue to grow. So in this first one, this was before, you know, we were trying to just get some things out and test them. Initially, what we had worked with is there was a researcher at MSU who had students that were collecting different plants and looking for hyperphosphorus absorbing plants. And those are what we wanted to get in here initially. I don't think that got completed, and so at that point we were just looking at, well, what else could we do? We talked with the local plant dealer who had these wetland plants and tried to get different characteristics of those. And we didn't have a whole lot of information about performance of these, but we were trying to see how well would they do in a fully saturated, you know, hydroponic type of system, and have wet and dry times. So that was a little bit of what we did, is just talking to them about hardiness of the plants, and then we'll see once we get the final results, maybe probably here at the end of October, November, we'll pull these plants and then measure the phosphorus in those, and we'll start to get an idea. We'll continue to rotate, so we'll take probably the best two, both from a phosphorus standpoint and from a, you know, resiliency standpoint, and put those in, and then maybe add 2 more into the mix, and we'll keep doing that each year, and as we add more and more mats.
Good afternoon, everybody. Thanks for joining to get an update on this wetland work. We've now, after this video that was put together last fall, we started a full year of collecting samples there, and so we can want to provide a few updates and then maybe talk about a couple things that weren't covered specifically in the video, and then we'll just go through a couple slides and then open this up to some questions that anybody might have. So there was significant plant growth that we had during the first year, and during that video you saw several different plants that we put out. We had Scirpus, Juncus, Carex, and Ranunculus were 4 plant species that we put out on the mats. I'll share some of the results of those, but interestingly, the first year we put those out in late spring, early summer, and when we harvested the tops off of those, you know, we saw some growth, but it was the plants when we left them out over the full winter, and then they started growing from early spring, and then we came out again this summer, that there was a significant amount of growth. The plants really did very well. We saw which species were doing better than some of the other ones. I'll show some pictures. We removed the ranunculus, which was not doing very well, and we added another mat of sweet flag, which is a new plant that we're bringing into the pond, and so we'll see how that does, and we'll continue to rotate and keep the better plants that are performing well in that system. So if we look at what the plant growth for wet and dry weights looked like, our wet weight on the left, you can see in grams what our average starting weight was for wet weight for the different plants at different dates. So this was, the blue here is at the beginning. We took some again later on in the year, toward the fall, when we did our some of our harvest of the tops of the plants, and then when we came back out, the average weight for this summer, after they've been out for a full year, was significantly better. Scirpus and Juncus probably performed the best. And again, this ranunculus here, not quite as good. We had about 100%, I think, of the Scirpus and nearly 100% of the Juncus that survived all winter long and then flourished in the spring. And we lost a significant amount of the ranunculus. We look at the dry weight, and again, just see something similar. This is after we pull the plants out, and we'll dry those and weigh those materials. And then we're also taking, in the process of taking tissue samples from these plants as well, so that we're looking at nitrogen, phosphorus in those plants through the sampling we're doing there. First year growth, again here, laying those out, and then that's for wet weight, and then the dry weight growth over here. You can start to see actually the Juncus had a little bit more dry weight growth than the Scirpus, but both of those plants did very well. Oops. So here's some pictures just to give you an idea. We had, that's about the average size of the Scirpus that we collected this summer, I think in June timeframe. So those were put out the previous summer. They went all through winter. We just harvested the tops and left the root mass into the mat. And then this is what we found when we came back out. The shoots on those were probably close to 4 feet. They were about 45 inches, and the roots were a little bit over 3 feet at about 39 inches. And the picture on the right, we just laid out the 4 of the plants, ranunculus on the left, carex, then Juncus and Scirpus on the right, just to kind of give you an idea of the shoots and root comparison between those. So they've done very well in those systems. Didn't talk a lot about the layout for sampling. So this is actually a slide I have from a different project, but describes the situation pretty close to what we have there. So from the field, we have a subsurface drain that is coming into our pond. And at our wetland, we have 3 drainage control structures that are set up there. The only difference is in this drawing, the automatic sampler is set up with a separate housing. In ours, the housing for the sampling unit is actually over top of the drainage control structures. So when you go inside of those walk-in structures, the drainage control structures inside of them. And essentially what that does is water goes over and builds up and goes over a V-notch weir that's in this system, and then we're able to measure the water flow at different points on that V-notch weir, and that allows us to run calculations to estimate the amount of flow that's coming through that system. And then we have an automatic sampler that's flow paced, so as a certain amount of flow goes through the system, it draws a sample, and then we collect those samples and do our analysis on those to look at what's the nutrients coming into the system, and then what's the nutrients in the water after it leaves the first pond, and then after it leaves the second pond. Some of that equipment looks a little bit like this. We have a transducer that sits down in the water that helps us to understand the stage or the height of the water in that structure as it goes over the V-notch weir. We also have low-cost water level sensors that we've been developing that we put into the units that also can read that information. And then we have those over the pond that will help us estimate the pond levels, so we can look at evaporation and other things with the water level fluctuations. And this is just if you're looking down inside one of those drainage control structures as water is going over top of that V-notch weir, just to kind of give you an idea what that looks like. So if we look at our water sample analysis for pre and post treatment in pond 1, so pond 1 is the one that has the floating wetland vegetation in it. we look at total pounds on a monthly basis. And pretty much across all of our months, we see a reduction coming out of that first cell. And this is, again, total phosphorus measurements that we've had. So we look at about annually right now, we're looking at about a 70% reduction. This is our first full year of data collection from October to October, which is our water year. And we still have very preliminary data. We still have a lot of things that we have to account for. I'll talk a little bit about some of those. So we can't specifically say that everything is taken up from the mats yet. We're still trying to understand the system a little bit better so that we can report out more accurately. But as to date, this is what we have been seeing so far. So again, as I mentioned, the performance, about 70% reduction in total phosphorus loading from what enters the pond and then from what leaves that first pond. We also have seen reductions both in dissolved reactive phosphorus and total phosphorus concentrations across the months. And we have had several algal blooms that have kind of come and gone in that first pond. And we know that those are accounting for some removal of nutrients. And so we're looking at some different models that we can try to estimate, what portion that has taken up. We have ordered, or in the process of ordering, 10 more floating mats that will go into that first pond. So that should cover a significant amount of the surface area there. And I think will help compete against some of the algae that was growing in that first pond. So for our next steps for the project, we'll be harvesting coming up here in November. We'll harvest the top of the plants. Those will get redistributed either out in the field or bring them to another place to recycle those. We'll do our tissue sample analysis of the plants so that we can look at exactly what the plants are taking up, not just what's coming out of the water. And then we'll be also looking at some additional measuring instruments to track water balance and flow calculations. So it's really important for us to have the flow calculations estimated correctly, because those are how we calculate loads. So we look at the concentration and the flow to get our loads for those, what's coming in and what's leaving. So we have some additional instrumentation we'd like to test and continue to tighten those down. And then we'll be looking at reopening that second pond. We've had some challenges in trying to figure out how to manage that pond appropriately to prevent it from clogging in the system. So we're looking at a couple different approaches and hope to have that finished up for this coming water year to collect data. So with that, I'll pause and we can open this up to some questions.
All right, so I'm going to begin reading our questions from the Q&A question box. Please just remember, if you do have a question for our speaker, to drop it in the Q&A box. Thank you. All right, first question. As we know, recently in point and non-point source pollution control through different emerging technologies has been applied. Did the Water Research Institute carry or plan to carry a study on quantifying floating low-end treatment performance in urban retention ponds, which, for our understanding, may remain elusive due to significant research gaps and/or poor studies?
So at the moment, we don't have any set up for urban retention ponds. We'd suspect there will be some similarities depending on how the ponds are set up, that as far as plant uptakes and other things that could translate over into an urban setting. But we currently don't have anything set up for an urban environment, just an agricultural at the moment.
Another question. Have you compared the results of this work to phosphorus filters, the filters that use the phosphorus absorbed material?
No, that's a great question. We have not done comparisons at this point, partially just because we're still, in the data collection process and don't have a final analysis. We have looked at similar wetland systems, and depending on the design and the holding time within the ponds, you get varying results between maybe 10 to 75% reduction in nutrients. And so what we've been seeing, at least preliminarily in this first year, matches up with what has been published for other types of systems, but we've not compared it to any phosphorus filters.
All right, what do you do with the particulate phosphorus that has settled in the bottom of the wetland?
So in our first system right now, part of what we want to understand is length of time for saturation in these. So our first system has the floating vegetation, and it acts a bit as a settling pond there. And then the second one has the filter media, which is kind of a sand, gravel, and limestone fine mix. Intent is for that first system to kind of drop out a number of the particulate phosphorus, sediment, and other things that might cause problems in the duration or longevity of the second pond. So we will leave those in and just continue to monitor to see how the system does and at what point we start to lose efficiency of that. And at that point, then we can pull those out from the first pond. We could remove those a couple different ways, but right now we just plan on leaving them in.
With the length of the roots by the end, was there any digging into the bottom that would impact the ability of the mats to float during higher flows?
These are all really good questions. So in our particular design, we put in a liner, a rubber liner, that so that we could try, from a research standpoint, we just didn't want anything that would be influencing from outside of the ponds for flow. So we put in a rubber lining. So we didn't have any problem even when the mats went all the way down during dry periods doing that. But we have talked with other researchers who have had that issue. If your water level goes too low, close to the bottom, and you have longer roots, they will attach in the bottom, and that will cause some issues for your floating mats. And as you saw in some of the pictures, the roots can get pretty long.
You mentioned that more mats will be added. Has an optimal level of plant coverage been determined at this point?
No, we've not done that. At this point, we've been adding a few mats at a time and trying to get an understanding of how they've been performing on a per mat basis, and then also looking at plant species. I think once we get a better idea of the plant species that we want to leave in there longer term and are performing better, we'll start, and as we're adding more mats, we'll start to get an idea of what that coverage area looks like. But we haven't done any estimates on those at this point.
Are the wetland plant species that are being tested ones that are native to Michigan?
Yes, we have worked with some local nurseries to get native Michigan plant species that we put out into our wetland, and that's what we'll continue to do as well.
All right, I'll give a couple more minutes just to see if anyone would like to submit some questions. All right, we have another question coming in. What do you envision the ideal size ratio of the land area drained versus your pond area?
So we had, I may have to come back to this question. So we had some initial ratios, I think, of about 1% that were some target numbers. And I need to double check these. We tried to go a little bit smaller than that, just to try to keep our since we were limited in land size and in trying to use these with producers, to go smaller than that. And I need to double check on that size that we have. I'm sorry, I should know that off the top of my head, but it's been a while since I did those calculations.
Did any of the plants survive the winter?
Yes, the majority of the plants all survived the winter. The certain plant species did better. The Scirpus, we had 100% of those. I think probably close to 90–95% of the Juncus, probably similar for Carex. And if I had to guess without looking at the exact numbers, I would say maybe 50% of the Ranunculus, 40 to 50% survived over the winter. But they clearly did not thrive in that hydroponic scenario that we had in those floating mats.
What are the soil characteristics or types in the watershed?
They're kind of loamy clays, where we are anyways, at least in the fields near us. Clay loams.
All right, I'll give a couple more minutes for any other questions. All right, Jeremiah, well, I don't see any other questions coming through our Q&A text box. So I just want to say thank you so much for what a wonderful and fantastic presentation that was. I just want to remind everyone else, a recording of this webinar will be available at the Michigan chapter of the Soil and Water Conservation Society's website, and also at the Institute of Water Research website. Thank you all very much for joining us today, and have a wonderful afternoon.
Thank you, Katie, and thank everybody for joining.